Yeah, thank you. Thank you for the opportunity for presenting our findings to this lake meeting. Lars and I represent a group of academic scientists who have uh, more than a decade of working with U.S. Environmental Protection Agency on all 5 Great Lakes. So we put this on the calendar to be able to report our findings for each year, and we try to get basically the samples from last summer done for this presentation for you guys. Lars has been very involved with the State of the Lake presentations and publications in the past decade. We just wanted to provide an update for this. So these we're going to go through lower trophic level indicators, mineral nutrients, TP and silica, also spring secchi and chlorophyll, also phytoplankton from UNREVI at University of Duluth, zooplankton from our group, as well as mysid shrimp, and then diporite and quagga mussels from the group at Buffalo State who do benthos. And yeah, today we're going to focus on the main basin. So most of our routine sites are in the main basin of Lake Huron. So we're putting this in the context of the last decade as well as long-term trends, i.e. going back to the 80s or as far back as we can, so 1998 for many of the trophic stuff, and then also compared to the other Great Lakes. So the story with TP, total phosphorus is a limiting nutrient for phytoplankton in freshwater. And like Lake Huron has been oligotrophic for a long time. In the mid 80s, it was up to 6 micrograms per liter and has steadily decreased, particularly around 2003, 2004. There has been some trace of recent increase, but this is still low concentrations of mineral phosphorus. Now, spring silica we use as kind of an index of the spring diatoms. So if you have increasing silica, you basically have fewer diatoms that are uptaking that. And generally, we have seen an increase of this spring silica since the same time period, this 2003 and 2004. We've seen pretty remarkable shifts in water clarity, particularly kind of shifting in this same time period of the early 2000s when mussels expanded. So we generally compare both spring and summer. There's kind of a 1980s regime with much lower water clarity than the current decade. And so what that leads to, this is showing all 5 Great Lakes for comparison. Some the blue box plots are basically the initial decade, and then the orange is the latest decade. You can see major increases in water clarity for Lake Huron, and currently it's the highest water clarity of any of the Great Lakes. Notice that Lake Superior really hasn't changed between those time periods, and again that just underlines the extensive power of the dry-seeded mussels. And if we look at phytoplankton, this is using satellite chlorophyll. We see, again, pretty sharp decrease in the spring bloom, which is the top values, the top line. And then also decreases in summer, but since in the past decade, really pretty stable values. It is very low. And again, yeah, it's stabilized since in the past decade. Now, UNREVI had the Duluth group started with EPA in 2007, and during COVID, he actually went back to some of the archive samples to push the time series back, and this was particularly important for Lake Huron. You can see that in this case, he's really pinned, and these are different phytoplankton groups. The browns are diatoms, which are important for spring blooms, and you can see the dramatic drop off of phytoplankton around 2003 and 4. both in the spring and the summer integrated sample. And this is the same for the southern basin. So there's 6 stations in the north, 6 stations in the south. But really consistent since that time period. When we look at zooplankton, the ones we've put blue dots on are basically the calanoids. They tend to be more deep water, particularly limnocalanus, the larger one, the dark blue one. There's also cyclopoid copepods in yellow, and then the surface dwellers like cladocerans, which are important, Daphnia, and other cladocerans that are important for grazers. And very similar to what we saw with phytoplankton, there was a pretty sharp decrease in the whole water column zooplankton biomass around 2003. And notice that this led to a major loss of Daphnia, the red, and also other cladocerans. These tend again to be in the surface. So we're seeing kind of a sudden drop in 2003, 2004, and very consistent since then, dominance by calanoid copepods. Turning to mysids, we do have really nice long-term data for mysids, and they're important in one of the largest zooplankton, or the largest probably in the Great Lakes, and an important food item, particularly for juvenile lake trout and other fish. We really benefited from the expanded data sets that the USGS, Tim O'Brien, provided, but you can see the density of mysids on the right has been very consistent. However, it is very low relative to the other Great Lakes, which is shown on the left. So that's divided into 2 time periods again. So you see things like Huron has basically had the same concentration of mysids over these 2 time periods. Michigan has actually dramatically decreased recently, and you probably saw Steve Pothoven's presentation yesterday on some exciting things for looking at that. Whereas Lake Ontario and Lake Superior tend to have the highest mysid abundances. One reason why Lake Huron has such low abundances, we believe, is because it's a relatively shallow lake with high water clarity, and mysids really like dark conditions, so they may not have a daytime dark refuge. So the husband-wife team of Alexander Karatayev and Luba Berlikova out of Buffalo State has done intensive surveys of quagga mussels in all 5 Great Lakes. In 2022, they did a lake-wide survey as part of the CSMI program. And then in general, many of the densities are now declining. These are different depth intervals, and then the bottom one is lake-wide. You can see that they're starting to reach a point where they're stabilizing. So a really neat thing that has continued from Tom Nalepa's work for Lake Michigan and Lake Huron. These are heat maps basically showing the expansion of quagga mussels on the top, whereas 2017 and 22 are relatively still high densities. And then the bottom has the native amphipod Diparia, which started declining in the early 2000s, and you can see is very sadly just continued to decrease. Lake Superior pretty much continues to be the only established Diparia population in the 5 Great Lakes right now. So if we look at all of these indicators, going from mineral nutrients to phytoplankton to zooplankton to mysids to mussels, all of these have either increased or decreased, but the red indicates basically a trend for lower productivity. So again, this is over the full time series, so the 30 years. But if we look at the most recent 10 years, we see very little change in these things, except for some increase in total phosphorus, which is colored blue to indicate some kind of increase in production. And then, of course, diapauria continues to decrease, and quagga mussels seem to be starting to stabilize or decrease. So not a lot of new things to report, but pretty much the status quo is continuing since that dramatic decline in 2003. So these are the take-home conclusions. Lake Huron has become more oligotrophic, so ultra-oligotrophic almost, and it's converged towards the state of Lake Superior and probably surpassed it. Also, but there has been very little change since 2005 in these open lake conditions. And we do think mussels are likely the most important drivers of this change. And of course, diapauria continues to decline, but mussel abundance is still very high. We do want to point out that these are only results from the main basin, and we do have Georgian Bay, North Channel, Saginaw Bay not mentioned in this talk, and we're working with collaborators and the Canadians at Ontario Ministry of Natural Resources and also DFO to try to beef up that data to get to you. So yeah, thank you very much, and we'd just like to acknowledge our partners.